first Camp Sunshine trip, so this is really great. I was proud to make it before dark yesterday and not get too lost, so that was really good. And Evan mentioned that Boston is, he said that we're the best. As a Texan, I find that really hard to state, but it's definitely not the best in barbecue, I can say. So, no, it's a real pleasure to be here, and I think I've met several of the families before. And so I think my goal today is kind of to tell you a little bit about something you don't hear too much about. I think Amy sort of led into that a little bit, which is both the pathology and the diagnostics and this new thing called precision medicine that you may have heard even the president talking about. And I think it's an area which really is important for your kids. It's really an area for pediatric brain tumors where these types of things have really had some of the best impact and have a lot more potential, like you've heard about, to impact them in the future and improve their lives. So I have a good set of things to talk about, but to be really honest, we have about 30 minutes talk or something. I want you guys to interrupt and focus on the things that you like, because I can go on forever. But it really is about going ahead and asking the questions that you have. And we'll have a little bit of time, I'm told by Amy, after, and I'm here until about 12:30 or 1 after lunch, so I'm happy to talk to anyone about almost anything. So let's get started. If I can run it like Amy said, there's lots of button choices, but none seem to activate. So what was the trick?
AJ.
I pressed all but 2 of them now.
Tricky sliding.
Okay,
It's really stumping some pretty smart people.
I'm just not strong enough. I won't fit the stereotype of pathologist, but I wasn't strong enough to press the button. What is precision medicine? And I think really there's a lot of confusion around this simply because of words, and we keep changing the words that we're talking about. And so it's important to keep up with words, as I think you all know as parents and going to the clinic and things, because it's what everybody is using to communicate, but it's not always big changes really in what's happening. It's more just the terminology. So the terminology that's changing now is that people are excited, all of us, because there are all these new tests and genomics and things that help to better define tumors. And so people have come up with new words. So precision medicine is the fashionable thing to use right now. You may remember when it was called personalized medicine. There was, I think, no real reason to change that, but the government decided that personalized sounded like a privacy risk and things like this. So the rumor was it got changed overnight on everyone from this slight sort of detail. And then genomic medicine is another word, and it really does come from the fact that right now pathologists are doing lots of diagnostic testing, and much of it now is able to be based on genomics or genetics tests or epigenetic tests. And so it's really these tests and the technology that have really pushed things forward as far as what might be useful to do as far as determining what type of tumor someone has. And this applies to kids as well as adults. And it's really to get that, why is it precision? Well, it's customized to your individual tumor. And what we were talking about before, and Evan was referring to, were giant trials. These are huge populations. I mean, so there's 3,000 patients planned to be in that trial that he mentioned. And so they're going to come up with a conclusion, hopefully, from that trial. That of those 3,000 people, some of them did better. The problem is it's not you. They're not going to point to your kid and say, your kid is able to do better based on results of giant population studies necessarily. And so what we're trying to do now is boil it down to predicting what's going to happen to you or your child when you go see the physician with much more precision, because right now it's a statistical association and it really is very imprecise by definition and depending on what the statistics show. This is examples of what we're talking about. So in the past, you would say, well, there's an organ that has a tumor or lump in it. Is it the brain or is it your skin? So that's precise. It's telling you the organ that the tumor occurs in. But we can also do that by age, pediatric versus adult. You can split it by low versus high grade. That one has a lot of important implications. Type, glioma versus pilocytic astrocytoma. I think we all agree, knowing that you have something more specific than just a glioma is important. And what we're talking about now is moving into an era where genomics help us say things like, instead of just glioma, it's a pilocytic astrocytoma. It's WHO grade 1. It has BRAF fusion, but it actually has a very specific BRAF fusion out of the 20 BRAF fusions of those two genes already seen. So you can already see that the data is starting to pile up, and that's unique. Really, we're talking about just in the last 5 to 10 years, and many times now, still just getting the one-word glioma or maybe low-grade glioma. So this is a big change, and it's a big change for the patient as well as for the doctors having to take care of the patient, because now there's all this information and you have to know what to do with it. And so the Precision Medicine Initiative is something that's going on with or without the government, depending on your view of government. Sometimes they're just taking credit for what's going on, but I think it's great that there's a lot of focus on it. So the White House has started this initiative. There's also the Moonshots Initiative, which somewhat overlaps with some of the goals of the Precision Medicine Initiatives. And it's really to try and help fund and add more money into the pot for doing these types of things. When you hear about precision medicine, cancer and tumors are the sort of headliner event for that, because many of the other sort of diseases don't have those types of associations that precision medicine can really help with quite yet. And so much of the money will go to cancer, but not all of it. And brain tumors are actually really well represented in many of these initiatives, thanks to people like you and other people with adult cancers of the brain. Many of the sort of steering committees and the physicians involved in these designing these strategies actually are directly very focused on neuro-oncology and brain tumors. So that's an important thing, but I think still it would be important to keep pushing for more and more of this to focus on what we have. So just want to make sure that people understand that medicine has always been sort of personalized or try to be precise, but it's really the focus and intensity that's gotten people excited, and the potential to change lives with things like these trials and the ability to now understand the disease at a level that really we just had no idea before. This was a picture of a bill. So in 2007, when I was searching for this talk, I found this bill that actually was sponsored by Obama before he was actually president, which was a precision medicine initiative. And one thing I thought was interesting is one of the first things mentioned is that it is focused on diagnosis. And I think that really gets lost currently in a lot of the sort of initial stages and excitement about what could happen. What's happening now is diagnosis. So I think that's partly what we'll talk about today. The new research findings are coming at a quick pace. I mean, I think you go to these meetings like ISPNO, and you realize that there's just a ton of activity out there. And again, it can feel a little different when you go and you're in the clinic and you're facing not thousands of genes and options available to you, but for us as researchers and as doctors looking at the research, it is really quite rapid. I mean, if you think about the progress in pediatrics with low-grade gliomas up until about 2007, it was really slow. We have that chart, and you're talking about decades going by without anything at all being new to talk about. Now it's very different. Every time you go into clinic, probably, there's going to be a lot of new information. Again, not all of it immediately actionable. And we're talking about decision-making is important to you, but this stuff eventually does change lives. So it's really important to think about every 6 months. There's papers coming out. There are really large efforts, I think, that are changing our understanding of the disease. It's about, often less than a year now even can happen that you have a new gene or a new mutation, and it turns into a diagnostic that might start affecting you now, because now you know something about your tumor that you didn't know before. And that can happen even overnight, because if we go to a meeting and I hear about a gene, now we have tests that are so broad that they may already contain the information. We just have to mine it for that new event. So that's a really important change. But really what it's about is this clinical care decision making. And so patients increasingly, as we're going to see, are responding to therapies. They may not be organized conclusions to that yet, but we're seeing things that we haven't seen before. So that really makes things go much faster, both for understanding what drugs give, as well as just how patients will survive after treatments that are standard even. And so new trials and drugs are now commonplace. I think everybody's pretty aware that one reason it's exciting to me in the sort of tumor and cancer space is that there is a lot of industry interest. So you have people like Evan working hard behind the scenes, even in for-profit companies potentially or non-for-profit companies who are interested in solving these problems, and that's not always the case. So I think the interest is there. I think, Amy pointed out sometimes it's focused on adult tumors, and so it's our job to kind of have this research and excitement, explain it to them, and then as advocacy groups talk to them about migrating that towards and applying those resources towards the children's tumors. And so that's happening, but it can certainly happen even faster. And I think what we're not used to is needing the level of new teamwork, both with you all as well as with our clinical colleagues and researchers. There's just a ton of communication and support that needs to happen, and money. So even though money is not the overall focus, I think I'm not standing here actually, except for the fact that you guys funded and made it sort of very attractive for us to be able to pursue our dreams of what we can do in the research arena. And so it's really hard. I think the best way to think about it is every researcher is a startup company. Everybody who's a PI running a lab doesn't have enough money to pursue the ideas they have and the things they want to get done. And so it's just like a company. You're very nervous about it. You have to figure out how to fund and employ people. I have people in my laboratory who have to feed their kids and do things and live their lives and have a good life. And so it's really nerve-wracking to run a lab, especially if you don't have that support coming. And as we know, the government doesn't provide sort of regular support of the level needed to achieve the goals that you and me have. So I think that's really critical to understand. And it's really important to do this through teamwork. So how do pathologists kind of diagnose kids and deliver precision medicine? Amy really pointed out that most people have not a great knowledge of what's going on behind the scenes. Pathologists are somewhat to blame for that. I was talking to her and I said, most pathologists are kind of not in the past super anxious to interact. They kind of hide from people, and they're the behind-the-scenes infrastructure. But if you think about it, what we're doing is actually the first step in your entire journey. So it's really a huge step. It's really an important determination that's not thought about a lot. And we've often let people just explain to you what's going on, who do not sort of generate the information and then discuss the reports with you. The level of detail and reporting and technology and assessments now going on make that really difficult. So what we've noticed is that we need to have more direct conversations with the families and the patients about what their testing is and what it implies, and work with the clinicians to really get you as much information as possible. So I think you're going to hear more and more about this in the future as well, both for children as well as adults. But to try and demystify some of it, I'm not sure how many people have actually seen their child's tumor under a microscope or a picture of it. So that's great. A few have, so maybe 3. So that's a pretty small percentage. How many have seen their MRI image of their child's tumor? Everybody. So this is something I think that's really important to know, that again, the MRI is very useful and helpful for monitoring, but it's not helpful and useful for the most part in getting that level of precision that we're now talking about. So if you think about it, we're all going to have a lot of work to do to just communicate more and try and close that gap. And this is what we're talking about. What the pathologist is doing behind the scenes is to give a diagnosis, which is a class. You've heard about subclasses and things like that. That's the first step. The second step is a tumor grade, and this is really kind of related to how aggressive the tumor looks under the microscope, or now with the other testing, what we think is the biology behind the tumor and how fast it's growing is probably the best way to think about that. So low-grade tumors, luckily, are growing fairly slowly compared to high-grade tumors. Treatment, response, and prognosis are all sort of serially linked to those prior steps. And so one thing to think about, and we deal with this all the time, and I think many of you may have as well, if we're not totally sure what the first step is or the diagnosis is, we can't give you very good information after that. And so sometimes we have to do that. We just can't tell what the tumor is, or we can't narrow the prediction of what's going to happen very well. And so that uncertainty is something you have to deal with. but we just keep trying to close that gap and make that smaller and smaller. We do that with all these different tools. And so the first one that we use every day is the microscope. And as Mark Kieran likes to point out, this is since the 1600s, so he kind of needles us a lot about our first step is to go back to the 1600s and look at your tumor. But you'd be surprised, the microscope is still an amazingly powerful tool and can still actually beat many of the things you've been hearing about and people bragging about they can do with genomics. They just don't like to talk about it. And they can't look at it because they didn't train in pathology, I'll say. But it's hard to train people. It's hard to actually, I went to school, I won't count how many years, and did training in pathology, and you get better every year for the most part until you start getting too old. So it's really useful, but it's not an efficient way to generate more and more information. So those tools that we have, like sequencing and RNA expression or methylation profiling, they're extremely helpful because they're more systematic and they can actually be done quicker and without training people for as many years. So anyway, we have a lot of testing that now can be employed to diagnose, and we'll see that some of that's very exciting and routinely implemented now. But until recently, most of this wasn't incorporated into that first step. We weren't really using it except as sort of more in between research and clinic and kind of trying to guess what would happen with a tumor. And there was a new change this year, just last month. The WHO, which probably some people have heard of, is the World Health Organization classification of brain tumors, just was updated this past few months, and there is a new focus on pediatric tumors, particularly like low-grade tumors have been described a little better in there. I will say that in my opinion, it still needs a lot of work. They did what they could, but it's a consensus book. When you have 73 authors, more than 19 countries involved, and all this information and focus, it's really hard for people to sort of come to consensus. So I think even Evan or someone said they were attempting consensus. That's kind of the idea, is that there's a rough consensus, but it doesn't really match what we know. So even though you have these guides, it's important to know that there's a lot more things that can be done, and at individual sites you'll have variation in what actually the knowledge base can be generated. But it's a great thing that it's been updated, and now it's much more aware of our interesting tumor types. But it's important to keep in mind, we have a lot of tumors, and many of these are pediatric brain tumors, and many of them are low-grade tumors. There's 128 brain tumor types in the book. I'd probably say at least half of them are very specific to young kids or young adults, and so that a lot of that diversity is coming from those different tumor types in children. And so it's a lot of work to have the same level of knowledge, about each of those, and they're very rare, many of them. So it's important to kind of keep in mind that it's not overnight that these things are going to change, and it takes a long time and a lot of effort on all our parts to organize all the patients that might be available for study and to learn more about them. And so we've done that a lot. I think this is when you look at the chart, you can see what a task that might be. Astrocytoma low grades, you've seen this before, about 30% of overall brain tumors, which is high, but then you start splitting it out into these different tumor types, which your kids may have, and it becomes smaller percentages. So I'd say most of the different diagnoses, except for say pilocytic, make up, maybe 5% is on average what you're talking about when you have these different diagnoses. Some are like 1% or 2%. And so when you just think about it from a researcher standpoint and a clinician standpoint, it's hard to get those predictions really tight if you're only seeing a small number of kids each year who have those tumors. So these large consortia or efforts to get people to work together are really important, that you all have funded and work towards. Gliomas and astrocytomas in kids, it's really important to understand that they are similar sometimes to adult tumors, but they don't overlap completely, and they have very different mechanisms of biology. That's why they're in the children, because they're different. They occur with different genetics as well. And so these are the tumor types that you see in sort of their order of incidence in children versus adults. And you'll see that the ones we're concerned about, low-grade glioma, are the top thing in children. These are very rare in adults, actually, and even though they're maybe second, the lower grade, they're actually completely different disease, even though they have similar names. And so And this is because they probably come from the developing child's brain. I think that's something that's really important to think about. They come from cells that are in the brain of the child that are developing, and some people talk about stem cells and other types of progenitor cells. And those cells are still growing because the kids' brains are still developing and growing. And so it's these mechanisms that are important for shutting off the growth that are broken most likely, and that's why they occur in children and not in adults with the same types of tumors. One thing that we've started noticing is that even though we talk about adults and children, age is a gradient. So, we all get older every hour, unfortunately. And so what we're seeing is that mutations and tumor types where we thought they were just associated with children are actually occurring in adults and vice versa. So this is really complicated, only been recently noticed when we kind of started doing more broad genomic profiling. It's still complicated because many laboratories are based around I study adult tumors or I study pediatric tumors. But like laboratories like ours and Farber, most of the people are kind of looking at both in the laboratory. And so then you start noticing that things like, try and hit the button for the light. Oh, I got it, great. You start noticing that things like histone mutations, which are in children shown here, are actually present mostly in kids under the age of 21. We're defining kids, depending on how your child acts. But you'll see, so there's very rarely over that age of 21, but there are some. And then many of the other mutations start to appear in a sort of more even distribution. So like TERT mutations are in both. PDGFRA is high in both. So these things really are important to keep in mind because your child also may have been 18 when they were discovered to have the tumor. That's probably a pediatric tumor, even though they may get older and turn into an adult. They still have a pediatric sort of type of disease. So I think it's important to keep in mind that there's complexity on this level. And the drug companies, I think this might help them a little bit in that it's not all one age group and it's not just kids, right, who have this disease. And so they can say, well, I'm not sure I want to take the risk of studying kids. It'll bleed over in adults as well. And so I think that's really an important thing to emphasize. Yes?
This might open a can of worms, so feel free to table it if it's too big of a question. But do we know anything about, if you look at a child's particular mutations, for example, and maybe other factors like the age they were diagnosed, do we know anything about likelihood of them getting a higher grade tumor down the line based on their...
Yeah, that's a really important question.
And does that impact potentially treatment?
Right. So fortunately, most children with low-grade gliomas do not go on to progress. And so in the adult types of gliomas, which are usually going to be IDH mutant gliomas, there's a risk that those may progress and have eventual progression to a higher grade and become a more aggressive tumor. I think even if it stays a lower-grade tumor in the adults, most of the patients have growth that just kind of continues, and so they may have a much longer period of survival in adults with IDH, but regardless of even whether it turns into a high grade or not, they still have a much more serious problem than most of the kids. But there is a small subset of children that we're trying to study right now, and many groups are focused on, which are how do we find the ones that are at more risk than the other kids for having that type of event. I'd say right now we don't have a great handle on that. There's some associations genetically that we've seen, but again, that's pretty bad information to tell someone if you're wrong about it. And so I think as a pathologist, I'm very aware of the impact of what I'm going to tell you, good news or bad news. And you don't want to be wrong very often. So people talk a lot about, say, CDKN2A as a gene that gets associated with things that go to high grade in adults or in children. But we don't yet know if you have that, what is that chance? Is it 100%? Is it 5%? Those are big differences. So I think that's the kind of thing that right now, again, it takes a lot of time and patience participating in research in order to better understand that and get people sort of numbers that kind of tell them more actual information for their actual child. And that's that precision aspect. Even if we get numbers, we're not sure for your kid unless we have a lot more understanding of how the tumor works. So that's a great question. Yes.
So as genetic information becomes more important with diagnosis and you find common mutations between say, brain tumors and other types of cancers, does that change how we understand not only the treatment, but just, well, obviously the treatment, but also just what that is, what brain tumor is, or like how to categorize it entirely? Right? Does that
I think there's a lot of talk about, well, we're going to diagnose everything by mutations, not to contradict you, but so there's a lot of talk.
Yes.
I think, it helps us think about it to talk about it, but I think the important thing is what it is, right? So context is super important. If you talk to Neil Rosen, who's been a collaborator of this foundation, he goes around, he's a big BRAF guy. He studied it in melanoma and other settings, and now in pediatric brain tumors. If you talk to him, the first thing he'll start blabbing about to you is that BRAF's great, but the context of the cell and where it came from is super important, and that's the most important thing. So that's the thing that kind of gets him excited, even though he spends all his time probably trying to find a drug to target these things, right? So I think it's important, I think, to understand the cell is kind of there that it came from. The cell is much more than one mutation. And so my feeling is that the context is going to be important for a long time because the mutations are going to be important, but there are many mutations that are actually the opposite of what you might think. They tell you that the drugs aren't going to work assuredly because the mutation makes it where the drug can't work. We're talking a lot, so there's a lot of sort of blurring of that line. Certain mutations mean the drug can't work because the drug isn't designed to work against that mutation. It was designed to work against another mutation. So there's a lot of complexities. I think the idea is that instead of generalizing too much, again, it's better to kind of deal with the whole situation, and that's hard because you have age, cell type, what type of tumor, what location it's in, and the mutations and the epigenetic state that's being described. All those probably are important. We don't know which one's the most important. We do know about action. So right now, the only action you can kind of take is if you find a mutation and there's a drug that's supposedly designed to match and help treat that, you need to match them. So the context is important for a lot of other things. But I think, it's important to kind of not get lost for all, the meetings are great. When I go to the meeting, it gets really distracting, though, a lot of the stuff is not action-based. And so that's important for researchers to do. In the clinic, sometimes they get distracted, too, by all the things that could be happening or could be going on. But if you, I think I've even heard from families, when you go to the clinic, the feeling is not there's thousands of choices for you and your child, right? And so I think that's the important thing to keep in mind, is that there are very few choices. And what we need is very important information as best you can about all the situation and variables that might affect you and your decision at that time point. And so that's the important thing to keep in mind.